In IB Bio, Transcription Translation Part 1, the focus will be on an overview of protein synthesis, in addition to some background information helpful in understanding transcription and translation. The essential ideas are genetic information in DNA can be accurately copied and can be translated to make the proteins needed by the cell. And secondly, information stored as a code in DNA is copied into mRNA. Here's the central dogma as presented by Watson and Crick, with the exception of RNA to DNA by the enzyme reverse transcriptase. The focus of this unit is the synthesis of RNA from a DNA template and the synthesis of a polypeptide protein from an RNA template. This part 1 of the unit presents an overview of the biochemistry of protein synthesis as well as some important background information. Here is another view of the central dogma, however it provides other key information. DNA is a two-stranded molecule, while RNA is a single-stranded molecule. Replication is the synthesis of DNA from a DNA template. Transcription is the synthesis of RNA from a DNA template. And translation is the synthesis of a polypeptide from an RNA template. All three of these processes use enzymes specific to the process. Here are three relevant IB syllabus statements. Define the term gene, state that a gene occupies a specific position on a chromosome. Eukaryotic chromosomes are linear DNA molecules associated with histone protein. Genetic information is stored as a sequence of nucleotide bases, A, T, C, and G, that code for the synthesis of a polypeptide. And chromosomes, depending on their size, have hundreds, if not thousands, of genes. While the concept of one gene coding for the synthesis of one polypeptide seems simple enough, there are numerous details to account for. Stay tuned. So a gene is a discrete unit of heredity consisting of a specific nucleotide sequence in DNA, which is transcribed into mRNA and then translated into a single polypeptide sequence that through the interactions with other genes or other gene products controls the development of hereditary characteristics, controls the development of our visible appearance, the phenotype. Now, one additional thing to consider when working to understand the one gene, one polypeptide idea is that some genes code for ribosomal RNA or transfer RNA, and these are codes which are not directly used as the template for a polypeptide. Transcription, step 1, takes place within the nucleus of eukaryotic cells. Remember that in prokaryotic cells, transcription would take place in the cytoplasm. The raw mRNA transcript is modified. This is step 2. The mature RNA transcript leaves the nucleus, and translation, step 3, occurs in the cytoplasm. The polypeptide is modified or joined by other polypeptides, remember hemoglobin with 4 polypeptide chains, to become functional protein. That's step 4. So, while the one gene, one polypeptide idea is a good one, there are details under the surface. Keep this in mind. Again, here is an overview of the sequence of events. Sequence of DNA bases transcribed to RNA bases translated to a sequence of amino acids. While there are unique single-copy genes that are of high interest to scientists because they code for functioning polypeptides, there are sequences of bases in the genome of each of us that are repetitive, non-coding sequences, sequences that do not code for protein. But these repetitive, non-coding sequences are useful in DNA profiling for crime, paternity suits, or immigration. Here's our first IB syllabus statement of significance to this movie. Compare the structure of RNA and DNA, mention the names of sugars, bases, and the number of strands. DNA is composed of deoxyribonucleotides. By now, you should recognize the components of a nucleotide. The second carbon of the deoxyribo sugar does not have an oxygen, thus deoxyribonucleotide. RNA is composed of ribonucleotides. The second carbon on the ribose sugar has an oxygen. DNA is a double-stranded molecule and has the role of storing hereditary information and directing the synthesis of protein. RNA is a single-stranded molecule, and its single-strandedness allows RNA to assume various shapes. With flexibility in shape, RNA has various roles to play in the synthesis of protein, including messenger RNA, transfer RNA, and ribosomal RNA. As well, RNA can serve as an enzyme. Stay tuned. DNA bases include A, C, T, and G, while RNA has no thymine and instead uses a similar pyrimidine base known as uracil. So here's a table that compares RNA to DNA. You should see the differences and the similarities. Study the details of this slide. Here's the next important IB syllabus statement of this movie. Explain transcription, including the enzyme involved and the direction of synthesis. Transcription is the synthesis of a single-stranded RNA molecule using one strand of DNA as the template. You can see this here. This is a sequence of bases being transcribed. This is the gene. Each nucleotide of RNA is put into the RNA polymer according to base-pairing rules. Since RNA has no thymine, where the DNA strand has an adenine, the complementary base in RNA is uracil. When the DNA has cytosine, the complementary base in RNA is guanine. When the DNA is guanine, the complementary base in RNA is cytosine. And if the DNA image here had a thymine, then the complementary base on the RNA would be adenine. It's unfortunate this diagram doesn't show that. Now you may ask, or may notice, that 3 mRNA bases code for 1 amino acid, and you may wonder why this is so. Now in the last slide I mentioned that 3 RNA bases, a triplet, code for 1 amino acid, so I've already given away the answer to this question of how is the nucleotide code read, translated, given 4 bases and 20 amino acids. If the bases in RNA were read singly, that could only code for 4 amino acids. If the bases were read, translated 2 at a time, that's only 16 possible combinations of bases, 4 squared. But a doublet code works for only 16 amino acids, and there are 20. But if bases are read, translated 3 at a time, that's 64 possible combinations of bases, 4 cubed. A triplet code works for 64 amino acids, but of course there are only 20. So the code has some redundancy, sometimes known as degeneracy. We use a triplet code in mRNA, and that triplet code is called a codon. So once again, a triplet of RNA bases is known as a codon, and it's translated into 1 amino acid. These 12 RNA bases, 4 codons, translate into 4 amino acids in the polypeptide. The codon dictionary is well studied. We now know what each of the 64 possible triplet combinations of the 4 bases codes for. For example, GGG codes for glycine. GGA also codes for glycine. Remember, there are 64 triplet combinations, but only 20 amino acids. This is called redundancy, sometimes called degeneracy. UUU codes for phenylalanine. AUG codes for methionine. And 3 codons do not code for any amino acid. These are called the stop codons, AUA, AUG, and AGA. Stop codons bring translation to an end. Here's the relevant IB syllabus statement. Describe the genetic code in terms of codons composed of triplets of bases and the relationship between codons and amino acids in a polypeptide. Remember that most organisms use the same 20 amino acids in the genetic code, although there are some exceptions to the 20 amino acid rule. Here is the second type of codon dictionary. You locate each codon using this side, then this side, then this side. Remember, the term codon refers to RNA triplets of bases. The triplet code produces 64 possible combinations of bases for only 20 amino acids. So the code is redundant, as can be seen here, where these 4 codons all code for proline. These 6 codons all code for leucine. But the extra codons also provide for signaling. AUG is the start codon, and these codons are stop codons, bringing translation to an end. Using the codon dictionary on the previous slide, we can determine the amino acid sequence from the given RNA sequence. UUU codes for phenylalanine, GGC for glycine, CGU for arginine, and UUU for phenylalanine. UUU always codes for phenylalanine. There is no ambiguity, and this diagram does a nice job of representing phenylalanine as a unique amino acid, a square, and putting the square shape into place wherever there is a UUU codon. So here's an IB syllabus statement that at first appears confusing, but with some thought it won't be difficult. Distinguish between the sense and antisense strands of DNA. The sense strand has the same base sequence as the mRNA with uracil instead of thymine. The antisense strand is transcribed. So here is the template strand of DNA. This is what's known as the antisense strand. The strand of DNA complementary to the antisense strand is known as the sense strand. Hang in there. This strand of DNA is the antisense strand. It is the template for RNA synthesis. It's the template for transcription. Maybe this slide will help you see it. This DNA strand is the template strand. This is the antisense strand. The sense strand has the exact same code as the RNA, with the exception of uracil instead of thymine in the RNA. Take a look. The sense strand has the same code as the RNA transcribed from the antisense strand. Based on the mRNA given in red showing the codons, which strand of DNA is the template strand, which is the antisense strand, which is the sense strand? You can turn the movie off now or follow along with me. This strand is the template strand. This is the antisense strand used to transcribe the mRNA. This is the sense strand because it has the exact same sequence as the mRNA with the exception of uracil on the mRNA instead of thymine. Hopefully you have a sense of this somewhat confusing terminology. If you're not sure, see me in class or go back and review this relatively short stretch of slides. Once again, the bases of the antisense strand of DNA are transcribed into complementary RNA bases. The bases of the sense strand mirror the bases of the RNA exactly, except that RNA has uracil instead of thymine. So as we move forward to look at transcription in more detail, we must remember that in a similar manner to replication, the synthesis of RNA involves nucleoside triphosphates. Two phosphates are cleaved off to form the nucleotide. The energy found within the bonds between the phosphates provides the energy for the anabolic synthesis of the RNA polymer. Here is the nucleotide to be added to the growing RNA polymer as a part of transcription. As we move forward, we'll be working on this IB syllabus statement. State that transcription takes place in a 5-to-3 direction. And in this diagram, we can see the RNA polymer with the nucleotides in a sequence identified by their bases. You can see the antisense strand of the DNA, which provided the template for the transcription of the complementary RNA base sequence. Lastly, it's important to note that the RNA synthesis took place in a 5-to-3 direction. The RNA molecule was synthesized in this direction using the DNA strand here as the template. The DNA template strand was oriented anti-parallel 3-5 relative to the 5-3 direction of synthesis in the making of the new mRNA molecule. Hopefully this strikes you as beautiful. And that brings us to the end of IB Bio Transcription Translation Part 1.